Okay, so welcome again to part 2 of the module 3. Okay, in this particular video, we are going to look at how mRNA is degraded. Okay, among other things also, but that's where we are going to start from. So relatively to rRNA and tRNA, mRNA is actually unstable, okay, as compared to the other two. And in part, we can attribute this instability to the action of ribonucleases. The action of ribonucleases is not the only reason why mRNAs are unstable. Okay, and ribonucleases are examples of endonucleases. Okay, so these are like endonucleases that only attack or that only cleave RNAs. So mRNAs actually exhibit wide range of half-lives. Okay, and of course, all mRNAs can't have the same half-lives. If it was so, then there will be a particular point in time where there will be no mRNAs in the cell. So different mRNAs have different half-lives. Okay, and these mRNA specific half-lives are controlled by specific sequences or structures within the RNAs or mRNAs themselves. So this is like a real life example of it be your own niggas, like wow for the best nature also. So there are structures or sequences within the mRNA sequences that will be like, Charlie, our time is up, we have to be degraded. Okay, so the differential mRNA stability is an important contributor, blah 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 blah. So let's talk about this point, pretty important. So the abundance of specific mRNAs in a cell is actually as a result of two things. Okay, the rate of synthesis and the rate of degradation. So if the rate of synthesis is higher than the rate of degradation, obviously mRNA levels is going to be higher. Okay, or it will be increasing. But if the rate of degradation is higher than the rate of synthesis, then the levels of mRNA will be reducing over time. But if these two are in steady states, okay, the parameters will be the mRNA levels will remain constant. Okay, so also the protein spectrum of a particular cell is a reflection, not the only thing, but it's largely a reflection of the abundance of mRNA template. And we know mRNA template go on to be translated into proteins. Okay, so if you go into a cell, the protein content or the protein spectrum of that cell is a reflection of its mRNA abundance. Okay, so now let's look at specific sequence, some of these specific sequences within mRNA. Okay, that are the snitches. Let me call them the snitches. Okay, so we have, but to be frank, the second one is not a snitch, right? So there are three elements, and these are cis elements. If we say cis, that means they are within the strand and they act on that same strand. Okay, they can't. So if two mRNAs are lying side by side, the one sequence molecule A cannot act on molecule B. If it can act on molecule B, that will be trans acting. Okay, but these elements are cis acting, so they can only affect the strand or molecule they found themselves inside. Okay, so these sequences are cis elements, and they are actually two. Okay, but there's a third one, and that one falls under the first one. So the first one is destabilizing element. Okay, and the second is stabilizing element. From their names, you can obviously tell what they do. Okay, the third one is AU rich elements or Rs. Okay, and these are examples, or they fall under the destabilizing element. So the stabilizing elements are the snitches. All right, they will facilitate decay, and stabilizing agents or a stabilizing element actually slows down decay or makes mRNA molecules more stable. Okay, so if you look at this diagram over here, the first one will be like an mRNA with a destabilizing element. Okay, so within the mRNA sequence, this destabilizing element is actually part of its sequence. So if this part of the mRNA is supposed to code for a protein, this part, the DE, is part of the sequence coding for a protein. But at some point in time, who Judas. So you see, Judas was actually part of Jesus Christ's disciples. Okay, but when the time came for him to, it's actually appointed. So no matter what happens, your true colors will be revealed. Right. So it's actually part of the mRNA. It's not like isolated, that it doesn't take part in protein synthesis or gene expression. It takes part, but it also plays another role, which is snitching on the mRNA. Okay, and recruiting the degradation machinery, right? And there are other molecules, mRNA molecules with stabilizing elements, okay, and these stabilizing elements are going to block the binding of the degradation machinery, okay, and hence stabilizing the mRNA. And the AU-rich elements are mostly in mammals, okay, and they are bound by a variety of proteins. So let's see, in the AU-rich regions, as I said, they bind to proteins. So the proteins that bind to the AU regions actually interact with the AU-rich regions, and these proteins form part of a complex called the degradation machinery. Okay, so the component of the degradation machinery are the exosome, the adenylase, and the capping enzyme. So all these are like individual proteins or enzymes, but they form a complex called the degradation machinery. Okay, and they interact with the R, or the AU-rich region, AU-rich element. Okay, so the degradation machinery is what is recruited onto the site, and that's the degradation of the mRNA. Okay, so for the stabilizing element, they occur in highly stable RNAs, and it makes sense. mRNA degradation rate can be altered, and this is in response to a signal. Okay, so let's look at degradation of mRNA in prokaryotes. So in prokaryotes, one thing I must say first before I begin with is, in eukaryotes, addition of polyase to the mRNA strand is it's like a signal to end transcription. Okay, but in prokaryotes, addition of poly A is a signal to start degradation. Okay, so one signal that facilitates degradation, prokaryotic mRNA degradation, is the addition of poly A tail at the 3' end. Okay, so you can see from here, 3' polyadenylation by this enzyme, it is generally a PN base, which is polynucleotide polymerase. But what nucleotide are we adding? The nucleotide is A, so we call it poly A polymerase. That's polyadenylase polymerase. Okay, so addition of the poly A tail facilitates degradation. Okay, so the initiation is actually by the removal of pyrophosphate from the 5' end. Okay, so this will be a facilitator, but what actually initiates the degradation in prokaryotes is, you see, the 5' is 5p, right? The phosphate phosphate group is actually found on the 5' end. Okay, so the phosphate is not actually a single phosphate, it's a triphosphate. So we remove 2 phosphates. Inorganic diphosphate is called a pyrophosphate. Okay, so we remove 2 of the phosphates, making the molecule a monophosphorylated molecule. Okay, and this monophosphorylated molecule will be like the substrate for degradation. Okay, so monophosphorylated mRNA will now be targeted by endonucleases. Okay, so endonucleases, in endo, that means they cut within the molecule. So they will cut within the molecule into small fragments. Then there will be 3' to 5' digestion of the small fragments that we got from the endonucleolytic cleavage, right? So the main degradation enzymes work as a complex. Okay, so the degradation machinery in prokaryotes is called the degradasome. Okay, and the degradasome consists or comprises of the RNase E, PNPs, a helicase, and another accessory enzyme. RNase E is the endonuclease. Okay, PNPs is the poly A polymerase. Helicase is what opens up the RNA strand, if just in case the RNA has formed secondary structures. Okay, so that's for prokaryotes. So let's see mRNA degradation in eukaryotes as well. For eukaryotes, there are numerous ways where our mRNAs can be degraded, but most of our mRNAs undergo degradation by one of 2 ways, and these 2 ways are deadenylation dependent. Deadenylation means the poly A tails have to be removed before the degradation will occur. That means they are deadenylation dependent. Okay, the other ones are some are deadenylation independent. Okay, but most of our mRNA undergo deadenylation dependent pathways. Okay, but and there are two of them, so we are going to look at the two. The first one is 5' to 3' decay deadenylation pathway, and the other one is 3' to 5' decay. Okay, so the first one, which is the degradation from 5' to 3', is of course it is deadenylation dependent. So the first thing that has to happen is deadenylation. And mind you, eukaryotes, we have poly A. Okay, so we have to remove the poly A as a signal of degradation. So the first thing that happens is deadenylation, which will be followed by decapping. You know the cap, which is the 7-methyl guanosine, the M7 gene. Okay, so the deadenylation occurs, the removal of the poly A's, then we will do decapping. The M7 gene will be removed. Then we will do 5' to 3' exonuclease digestion. Exonuclease means we start exo. That means we start from the ends, outer parts, not endo. Okay. So the enzyme that removes the cap is called the decapping enzyme, and it is made of two proteins, DCP1 and DCP2. I call them decap1, decap2. Okay. So this decap1, decap2 are the enzymes responsible for removing the M7 gene cap. Okay, so actually the decapping enzyme competes with an initiation complex for the 5' cap. You know, before we've not actually talked about this, but when translation is to begin, like before translation begins, there's something we call the translation initiation complex, and that recognizes the mRNA by binding to the cap. So in this case, the decapping enzyme competes with the translation initiation complex. Okay, so whichever will win is actually based on different signals. So if the mRNA is deadenylated, then obviously the decapping enzyme will win. Okay, so the decapping enzyme will remove the cap. Okay, and it will yield a monophosphorylated RNA end. So probably it also does removal of the pyrophosphate, making the mRNA molecule monophosphorylated. Okay, and also recruit or will serve as a substrate for an exonuclease called XRN1. Okay, and XRN1 will digest the mRNA molecule from 5' to 3'. Okay, the second pathway, or the second deadenylation dependent pathway, is 3' to 5' decay deadenylation dependent. So that one too, of course, deadenylation has to occur first, right? After occurring, because we are degrading from the 3' end to 5', we are not going to touch the 5' cap. We are going to degrade from the 3' end rather. So after removing the poly A's, an enzyme called the exosome, or the degradation machine called the exosome, that's like a ring structure with about 9 proteins, will degrade the mRNA from 3' to 5'. Okay, so this is it. And degradation may occur within discrete cytoplasmic particles called processing bodies. So if mRNAs are being degraded, mostly they occur in particles called processing bodies. Okay, so there are other degradation pathways for eukaryotic mRNAs. Okay, but these are for like specific mRNAs and not most of our mRNAs. Most of our mRNAs undergo one of these two degradations, but there are some few mRNAs that undergo one of these four we are here to talk about. And the first one is deadenylation independent decapping. So this one is similar to the first one. We are going to decap, but that one does not wait for the poly A tail to be removed. You see, in the first one, we made sure the poly A tail was removed before we touched the cap. That's why we call it deadenylation dependent, right? The next one, this one is independent. Okay, so this one too is 5' to 3', but poly A deadenylation independent. And there's another pathway called degradation of non-polyadenylated histone mRNAs. Hey. So there are some mRNAs, eukaryotic mRNAs, that do not have poly A tails. Okay, so obviously there should be another way of degrading these people or these mRNAs, because they don't have poly A tails. So a different way has to be employed. So in those histone mRNAs that lack the poly A tail, we add our own polynucleotide, and in this case we add in this case we add poly-U, okay, using poly-U polymerase. So we add poly-U, then that will facilitate the degradation. Another pathway is degradation of some mRNAs being initiated by sequence or structure specific endonucleotide. So this one is, there will be some sequences like the destabilizing enzymes, destabilizing element or other element within the molecules, the mRNA molecules themselves, okay, that will be recognized by specific endonucleases. So these are more direct. So there will be some sequence, there are some mRNAs that have specific sequences that an endonuclease can cut. So this one does not work with the capping or tailing or anything, just endonucleases coming to cleave it, to degrade it, okay. And some mRNAs will be targeted for degradation by microRNAs, okay. And this is like the one we did in when we talked about the risk, the Argonaut plus the risk blah 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 blah, right? So Argonaut plus some proteins will form and the risk protein will form an activated risk complex, then a microRNA will come in, but that one was siRNA, okay. So this is similar to that one as well. This is a summary, like a table summarizing the pathways we've actually talked about. So now let's talk about surveillance system, okay. The surveillance systems, there are two. We have two surveillance systems, and they are there to capture or to detect aberrant RNAs with abnormalities, okay. We have two surveillance systems, one in the nucleus and one in the cytoplasm. The one in the nucleus is called a nuclear surveillance system, and the other one in the cytoplasm is called a cytoplasmic surveillance system, okay. So let's talk about the nuclear one over here. So nuclear RNAs, that means RNAs in the nucleus that are aberrant, will be identified and destroyed by the NSS. NSS is nuclear surveillance system. And so the surveillance, how these aberrant RNAs are identified, involves identification and tagging. So if you're aberrant, you have to be identified first, then they will tag you, right? If you are tagged, that means you are bound to be destroyed, then obviously be distracted. Then the destroyer is called a nuclear exosome. So what actually does the destruction or the degradation is called a nuclear exosome. This is because nuclear means we are dealing with the NSS, the nuclear RNA surveillance system, right? And functions of the exosome is processing of some normal substrates of non-coding way, and also complete degradation, and we are focusing on the degradation of aberrant RNAs. An example of this system is in yeast, the TRAMP complex in yeast, okay. So the TRAMP complex in yeast will recruit an exosome to aberrant RNAs and facilitate 3' to 5' exonuclease degradation. It's not important. The second surveillance system is the cytoplasmic one, CSS. So this one is probably if the aberrant mRNA was able to escape the nuclear surveillance system, or probably it wasn't aberrant or it wasn't mutated when it was leaving the nucleus, but after leaving the nucleus or upon entering the cytoplasm, probably something happened, right? So the CSS will also be there to detect these mRNAs. But for the CSS, the cytoplasmic surveillance systems, there are 3 detection types or 3 defects that so let's say there are 3 different surveillance systems that is CSS employs to detect the aberrant DNAs. We have the NMD, let me mention by their names, the nonsense mediated decay, non-stop decay, and no-go decay. So one way of making you guys remember these things like easily is the no-go is something is blocking it, something is blocking something. I'll explain. The non-stop means there is no stop codon, so transcription will occur and like there will be no termination of transcription, so it is non-stop. And the nonsense is like premature termination, okay. So the mRNA we've coded doesn't make sense, okay. So let's look at the first one, the nonsense. You realize there will be, let's say there's an mRNA we are transcribing, and we are supposed to end somewhere here, but one sequence will be, let's say, I don't want to call it a codon, but some sequence will be mutated and obviously to be converted into a stop codon. Okay, so in that case, you are causing an NMD. So you are causing a nonsense mutation. Okay, you are prematurely terminating transcription, and that mRNA won't make sense because whatever is supposed to read it knows that it has to continue to some place, but there's no way I'm supposed to stop. That's why we call it nonsense mediated decay. Okay, so all mRNAs that are prematurely terminated are detected by the nonsense mediated decay. Okay, and all mRNAs that do not have stop codons, that means transcription will occur till those are detected by the non-stop decay. And those mRNAs that, let's say, when they were undergoing transcription, something happened and now the ribosome has been stalled, or there's a blockade on the way, so the RNA ribosome cannot proceed. Okay, so this is called no-go decay. So these mRNAs will be detected by the no-go decay pathway and they will be destroyed, right? So for the nonsense way, I've already said almost everything about it, but how these are targeted is the use of UPF proteins and SMG proteins. The UPF proteins we employ are UPF1, UPF2, and UPF3. The SMGs, pay attention, SMG1, 5, 6, and 7. Right? So 1, 5, 6, and 7. So these are the proteins that play a role in identifying or targeting mRNAs, the nonsense mRNAs, those with premature termination. Okay, so the recognition of the termination codon as premature involves unusual 5' 3' UTR structure. So mostly how they realize this particular mRNA is this particular mRNA has or was prematurely terminated is, you see, when an mRNA is supposed to, when mRNA is transcribed, you realize there is a 3' UTR, right, and a 5' UTR. So let's say this is the normal length of the 3' UTR, but here lies the case. This particular mRNA was stopped over here, so now you have a longer 3' UTR. Okay, so instead of it being from here to here, now you have from here to here. So when they realize the length of the UTR is abnormal, that's one main indication of, so one main indication that that particular mRNA might have been prematurely terminated. And also when there is a presence of downstream exon junction complexes. Okay, so these are called the EJCs. Okay, so when these are present in mammals, that also tells that that particular mRNA was prematurely terminated. Okay, and then they will be targeted by these proteins, either the UPF 1, 2, and 3, and the SMG 1, 5, 6, and 7 for the nonsense mediated decay. Okay. So this is the definition for the EJCs. They are protein complexes that assemble at exon junctions during splicing and assist in RNA transport, localization, and degradation. Okay. So the next one, non-stop decay. I've mentioned this, but they also employ a set of proteins called the ski proteins. Okay, so when the non-stop, the substrates, the mRNAs that do not have the stop codons, okay, they are generated mainly by, I think I've transcription, termination, in the nucleus, like in an I've said this. So the ribosome is released. So when the non-stop decay pathway senses or detects an mRNA with no stop codon or infirm stop codon, right, it uses the ski proteins, one of the ski proteins called the ski7 protein, to release the ribosome. Okay, because it's like car free, there is no break in it. Okay, so it will use the ski7 protein as a brake to remove the ribosome from moving further. And the no-go, I'm not sure there are any proteins associated with the no-go. Okay, and I've explained it already. So let's look at mRNA localization. So at this point, we've been able to transport it Well, is that the right assumption? Yes. So we've been able to transport the mRNA to the cytoplasm. Okay. All mutated or aberrant mRNAs have been, degraded, right? So at this point, we are dealing with only correct mRNAs. So how are these mRNAs localized? So mRNAs are going to be translated into proteins, and proteins have to be compartmentalized. Like they have to work at specific locations. So how are these mRNAs? And even some mRNAs have to be transported back into the nucleus. So how does the cell know that this particular mRNA has to go here, this one has to go here? Okay. So there are two. I don't know. Is it here? Okay, probably not here. Okay, there are some hypotheses that they made, or I should call them propositions, but I'm not sure it's here, so I won't mention them. So the assumption is probably they are translated in random locations. Okay, and these locations where they are translated will be determined by their point of entry into cytoplasm. So where from the nucleus, which part of the nucleus did they exit from, and where in the cytoplasm did they enter, and also the distance that they may have moved away from the nucleus. Okay, so probably these 2 points are going to determine where the mRNA is going to be translated, like randomly. Okay, but however, mRNAs are translated at only specific sites. As I said, they have to localize. That means they have to transport it to wherever they function specifically. Okay. This is important. This is an example of importance of localization. Okay. So as I said, in some cases as well, the mRNA itself has to be transported from another one cell to another cell. Okay, like in plant, for example, RNAs are exported or transported through the plasmodesmata for long distances. Okay, and they are transported via the phloem. Okay, so let's talk about the mechanisms for the localization. So these are 3 propositions made. Okay, one is the mRNA is uniformly distributed but degraded at all sites except the specific place of translation. Another one or assumption is mRNA is freely diffusible but becomes trapped at the site of translation. Okay, and the last one that makes the most sense is mRNA is actively transported to a site where it is translated. So we are not going to do any try and error. We expend energy, okay, to make sure the mRNA is transported to where it should be. We are not just going to transport it to everywhere, and the mRNA itself is going to find no. You're not doing that. So out of these 3 proposed or assumed mechanisms, I think this was mentioned to be the most sensible. So yes, that's why they say active transport is predominant. So I guess all these 3 actually happen, but the most active one is the last one. So the transport is actually achieved by these motor proteins. You know the cytoskeleton of the cell, right? You do advanced cell biology, so I'm sure you do. So the 3 motor proteins that they use, okay, are the dyneins and kinesins. Dyneins, kinesins, and myosins. The dyneins and kinesins travel along microtubules. Sorry, dyneins and kinesins travel along microtubules. Okay, and myosins travel along actin fibers. So these are how the active transport of the mRNA is done. Okay, so another one is RNA editing. RNA editing is just a phenomenon where the nucleotide sequence of a matured RNA is altered. You are just editing something. Okay, so whether you are adding, you are deleting, you are substituting, whatever you are doing to change the original sequence of an RNA is called RNA editing. Okay, so this is an example. So RNA editing occurs at individual bases. So in this case, we are actually looking at the molecular level, like the nucleotide level. Okay, making changes to the nucleotides, the bases themselves. Okay, not like removing chunks of genes or something. This is an example. I'm not going to bother with it. So let's look at this example. I think it was discussed in class, you realize in these are in different tissues. Okay, so the two scenarios I'm going to mention are in different tissues, but in the first one, this particular sequence of the same gene, right, has a sequence of CAA. Okay, and the CAA makes the mRNA encode for this large protein. Okay, and this large protein is found in liver and it has 4,563 residues. But the same mRNA is edited in a different tissue, like in the intestines, and that particular sequence of the mRNA is converted to UAA instead of CAA. UAA is a stop codon, so that means the protein is going to be shorter, okay, than the one in the liver. So you realize the protein that this one codes for is shorter than this, and this one will have like residues of 2,153. So that's like an implication of what RNA editing does. Okay, just an example. So we say RNA editing is directed by guide RNAs. Okay, so we use guide RNAs to direct RNA editing. Okay, so if you look at these diagrams, you realize let's say you have which one should I use first? So let's look at this one. You have your, this is pre-edited, the RNA you want to edit, right? Then use your guide RNAs. Then the guide RNAs will help you insert the RNAs you want at specific sites. Okay, so the guide RNAs are just helping us or directing us during RNA editing. Extensive RNA editing these are not important, and I feel like these things, when you are reading, you are going to understand these 2 points. So the last point, editing is catalyzed by an enzyme called editosome. Okay, editosome is a complex, a protein complex, and it comprises of an endonuclease, exonuclease, terminal uridyl transferase activity, and RNA ligase. You know what endonuclease does? Endonuclease cuts within molecules. Exonuclease cuts at the terminals or at the ends of molecules. Terminal uridyl transferase, that means that enzyme transfers a uridine, that's the U, but it transfers to the terminals, so it transfers to the outsides or the ends of the molecule. Okay, and the ligase joins molecule. So the endonuclease, exonuclease, the terminal uridyl transferase, and the ligase make up the editosome. Okay, I think we are actually done, not almost done. So I'll actually see you in module 4, the take home message. I'm sure if you read this will be like a summary of the whole thing, the entire lecture to you. Right, I'll see you guys.